The nervous system is composed of cells known as neurons. Neurons contain cell bodies, which are their centralized location that contain the nucleus and all their organelles. These cell bodies have extensions known as dendrites. These dendrites will receive messages from other neurons. Neurons send out messages via their axons, long, singular, tail-like structures. At the end of an axon is a synapse. A synapse contains synaptic vesicles that contain neurotransmitters, which act as the messages to other neurons. Now, let's take a closer look into how a neuron fires by examining its action phase. The first phase of action potential is known as resting potential. When the membrane of the axon is at the resting potential, most voltage-gated sodium channels are closed. Some potassium channels are open, but most voltage-gated potassium channels are closed. When a stimulus depolarizes the membrane, some gated sodium channels open, allowing more sodium to diffuse into the cell. The sodium inflow causes further depolarization, which opens still more gated sodium channels, allowing even more sodium to diffuse into the cell. Once the threshold is crossed, the positive feedback cycle rapidly brings the membrane close to equilibrium of sodium. This stage of the action potential is known as the rising phase. After the rising phase, voltage-gated sodium channels inactivate, halting the flow of any more sodium ions. After this, most voltage-gated potassium channels open, causing a rapid outflow of potassium. This stage is called the falling phase. In the final phase of the action potential, also known as undershoot, the membrane's permeability to potassium is higher than at rest, so the membrane's potential is closer to the equilibrium potassium than it is at the resting potential. The gated potassium channels eventually close, and the membrane's potential returns to the resting potential. Action potentials occur whenever a depolarization increases the membrane's voltage to a particular value, called the threshold.
When the threshold is reached, voltage-gated ion channels open, causing an influx of sodium ions to rush into the membrane. This rapid depolarization will spread, opening more voltage-gated ion channels, which will in turn carry the action potential across the membrane. Now let's take a look at what happens when an action potential reaches a synapse.
An action potential arrives, depolarizing the presynaptic membrane. The depolarization opens voltage-gated channels, triggering an influx of calcium ions. The elevated calcium ion concentration causes synaptic vesicles to fuse with the presynaptic membrane, releasing neurotransmitters into the synaptic cleft. The neurotransmitter binds to ligand-gated ion channels in the postsynaptic membrane. In this example, binding triggers opening, allowing sodium ions and potassium ions to diffuse through. In describing a synapse, we refer to the transmitting neuron as the presynaptic cell and the neuron, muscle, or gland cell that receives the signal as the postsynaptic cell.
A reduction in the magnitude of a membrane potential is also known as depolarization. In neurons, depolarization often involves gated sodium channels. If a stimulus causes gated sodium channels to open, the membrane's permeability to sodium increases. Sodium diffuses into the cell along its concentration gradient, causing a depolarization. An increase in the magnitude of a membrane's potential is also known as hyperpolarization. This makes the inside of a membrane more negative. This can result from any stimulus that increases the outflow of positive ions or the inflow of negative ions. The opposite charges across the plasma membrane is the source of potential energy. This charge difference or voltage is called the membrane potential. For a resting neuron, one that is not sending a signal, the membrane potential is called the resting potential and is typically between negative 60 and negative 80 millivolts.
The membrane potential is generated by the sodium-potassium pump, which transfers unequal numbers of cations across the membrane. It pumps 3 sodium ions outside of the cell while taking in only 2 potassium ions, which makes the inside of the cell more negative. In addition to this, there are potassium channels that freely let potassium ions diffuse across their concentration gradient and out of the cell, further taking away positive charge from inside the cell, making it more negative. Chlorine and sodium ions have a more difficult time crossing the membrane, which helps retain potential difference. Also, negative organic ions that reside in the cell cannot cross the membrane, which keeps the inside of the cell more negative and helps maintain the potential difference.